students, welcome to my third class of botany. In previous classes we study about binary fission, multiple fission, gamule, and today we are going to study about sporulation. Before going to study about sporulation, first I want to clear one thing, that what is your spore. It means gamete, and I hope you all know about gamete and what is it. Nextly, this process is mostly common in asexual reproduction, that means your sporulation process. This sporulation process is mostly common in asexual reproduction, as we are studying about your asexual reproduction, found in many species of your algae and fungi, and there are various types of spores like your Zoospores, Aplanospores, Hypnospores, and Chlamydospores, conidia. So at first we are going to study about your Zoospore. As the term denoted that spore means, you already know that it is your gamete, and zoo means your motile. Is that clear to everybody? Zoo means your motile and spore means your gamete. That means it is only the study of your motile gametes. So now I want to tell you something about your zoospore, that it is a motile and obviously haploid and thin walled spores produced in your sporangium. This is your sporangium. Actually, this red color part is your sporangium. It means that it is a single pocket which consists of a number of spores. That means it consists of a number of zoospores, and actually zoospores are these structures are zoospores, and it consists of flagella. These tentacles are your flagella. These are the flagellas. By the formation of flagellas, zoospores are able to stand by its favorable conditions. So they produce, because they are produced over unfavorable conditions. Actually, the haploid nuclei of zoosporangium divide mitotically. Previous stages are also some of your short notes I also tell about mitotic division. So it is also the parts of your mitotic division. Each nuclei get surrounded by some types of cytoplasm. It also consists of number of particles of your cytoplasm. After breaking these cells, breaking this sporangium, it goes out with some types of cytoplasm, because this is the cytoplasm nutrient mediums and this is also cytoplasm nutrient mediums. After break down the cell, it goes out and it's burst out, and cytoplasmic nutrient medium with the zoospore and flagellated structure comes out, as they are as it is a common process in your green algae and lower fungus. Each nuclei is gets surrounded a part of cytoplasm nutrient medium contains, and each sporangium consists of your flagella. Again I repeat you, and the flagellated structures lastly burst out from the cell, means burst out from the sporangium to outside of the spore, and it is all about your zoosporangium, which forms in mostly in green algae and lower fungi. It is the basic concept of your zoosporangium. Nextly we are going to study about your conidia. Then know what is conidia and how the structure is developed. Okay, conidia form, this is the structure of your conidia. and your conidia is consist of conidiophore and a number of conidias, and it is a hyphal joints. Before going to this structure, I also tell you the hyphal joints. What are the hyphal joints actually? This is your conidiophore, and these are actually it is these are your conidias. The first point of your note says that these are non-motile spores produced single or in chains. It is the outermost cell, outermost structure, because it is your projection, finger like projection is there, and it is these are the outer structures. The conidia are formed not inside this body, it is formed in the outside of the body, so it is outer structure, exogenous structure. Actually it is not endogenous structure. Exo means your outside, so these are the exogenous structure of conidias, which are placed maybe in single, maybe in double, maybe in triple, maybe many. It's also a chain also. So these are non-motile spores. Obviously these are non-motile spores because there is no formation of flagella is there. The cells are toe in structure, hard in structure. So these are non-motile spores produced single or by chain. Construction at the tip of hyphal branches called conidiophores. So these are your conidiophores. They are produced exogenously. Previously I told you that exogenously means outside. That means these are the outside structure. It is the total surrounding, and the spores are actually formed in the outside of this structure. So it is exogenous. Exo means your outside. Exo means your out. Exogenous and dispersed by wind, and when fallen, suitable substratum produces a new mycelium. When it is dispersed by wind and in the favorable condition, it is dispersed. And before going to another one, I also want to clear one word which is present in this conidia, that is your hypha. Consists of hyphal branches. Actually these are the hyphal branches. This is the branch, and these joints, a single joint and the another one. These two are joined in a single stage, and the another one. These are adjacently joined one to another, and formed a branch like structure. So these types of arrangement are otherwise known as your hyphal branches. Nothing to worry, this is your hyphal branches. So it is basically forms in your some types of fungal species, like your most commonly known your penicillium and Erisfe, etc. Some of the examples of your conidia. Nextly, we are going to study about your Chlamydospores, and in the next video we are going to study about your Chlamydospores. Spores are mainly consist of hyphae and Chlamydospores itself. So there is only two headings. Those are your Chlamydospores, which are toe and rounded in structure, and these are the conidial, these are the hyphal joints, which are conjugatively forms a rounded structure. This is your hyphae, and these Chlamydospores are actually thiel-walled and non-flagellated resting spores. That means spores are rest in condition in these Chlamydospores because the conditions are unfavorable in this time of formation of your Chlamydophore. when the hyphals are cell agium, which are rounded in shape, separated from each other and develop a thick wall. That means the spores, that the Chlamydomonas spores, which are rounded in structure, actually separated by these types of hyphae, which are separated. You look this diagram here. These are separated by hyphae. These are the hyphae structures, and the conidias are separated from the hyphae because it is the unfavorable condition. They store their reserve food materials and are capable of withstanding long unfavorable condition because it is apparently a toe and large structure above this, all these structures of your, you may call your conidia and other structures. In return of favorable conditions, these spores are germinated and produced to new mycelium. Most types of structures are also found in this Chlamydomonas. These are some types of examples of your Chlamydomonas. So these are some points about your Chlamydomonas. Nextly, we study about your Aplanospore and Hypnospore, and last one is your fragmentation process. So these are your Aplanospores, which are also non-motile in nature, and previously we studied that in Chlamydomonas rounded structure and thick walled, but here Aplanospores are non-motile but thin walled. You have to remember that it is thin walled. Haploid spores produced inside the sporangium. All the spores are apparently haploid in every short notes. They are produced under unfavorable conditions. The previous one also produced in unfavorable condition. It is also produced in unfavorable condition, and it consists of your vegetative cells. These are the vegetative cells. In every part of these cells, it produces some types of Aplanospores. These are the some nutrient medium of your Aplanospores, the Aplanospores stores their food and sometimes food materials and granules in this structure, or in this rounded, in this structure, which are vegetative cells, and shrinkage to develop a wall around to form an Aplanospore. They form a wall around it to under favorable conditions. They produce to spore germination and form new filaments, and mostly this type of formations are takes place in some species like your Spirogyra and Althrox, etc. So some basic things you have to remember in this concept, that all these structures are same, but in Aplanospores it is produced in unfavorable condition and mostly in thin walled. It is not thick walled, it is thin walled. So you have to remember one thing about that. This is your thin walled structure and mostly haploid cells. So nextly we are going to study about your Hypnospores. This is the structure of your Hypnospores actually. Hypnospores, it is mostly thick walled spores produced inside the cell. These are also produced inside the cell, and these are thick walls. Previously I told you about thin walled in your upper diagram of Aplanospore, but thin walled, but this is your thick walled. In this case of Chlamydospores, I already told you that these are also your thick walled spores, but they are rounded in structure. Under unfavorable conditions, Hypnospores are also produces shrinkage and separated from the cell wall. That means these parts of consisting of Hypnospores, these are actually the Hypnospores, and consist of some types of Spirogyra filaments are also present in here. These are your Spirogyra filaments, and these are your Hypnospores. These both Spirogyra filament and Hypnospores are studied in your Vaucaria and Chlamydomonas structures. So the Chlamydomonas structure and the Chlamydospores are quite different. So don't confused about that what is your chlamydomonas and what is your chlamydospore. Chlamydospore is a total extra structure, and your chlamydomonas is just an example. So Spirogyra filamented structure and Hypnospores are just like your Aplanospores, but in Aplanospores the wall is thin and genetic your nutrient medium and food material are stored inside the Aplanospore, formation of a wall in it. But in Hypnospores they are flagellated in structure and filamented in structure, and Hypnospores are developed in thick wall from non-motile Hypnospores. Why they are called non-motile? Because they are condition they produce their spores in your unfavorable conditions. Each spore gives rise to a new plant. So they are produced in, some are produced in thick wall and some are produced in thin wall. This is the basic of your Hypnospores. After that, I want to tell you something about your last but not the least, your fragmentation process. Fragmentation process means it is a simple process of reproduction which occurs in your algae and fungi basically. It is a process in which your thallus divided into many fractions. If you take that this is one type of organism and some types of algae or fungi, it may be look like that. Sometimes of algae and fungi structures are present. If this is your thallus structure, if the thallus break down some parts, then the other side fragmented part is liberated and gives rise to a new plant. This is just about the basic concept of your fragmentation process. Fragmentation means just breakdown, that's it. This my note is also tell that some only 2 points are there. The points are, it is a simple process of reproduction which are occurs in algae and fungi, and this process of thallus divided to into small fragments caused by your mechanical injury or some other type of dissolutions or middle lamella breakdown and gives to two plants. That means it's if it break down some types of mechanical injury that takes place in a single part, then this part is going to a new plant, and the remaining part is also form a new plant. Breakdown and formation of two new plants, this is simply called as your fragmentation. This is all about your asexual your sporulation process and some of the sporulation parts short notes. Next class we are going to study about your vegetative propagation and some of their types, natural vegetative propagation or artificial vegetative propagation. So stay then tuned. Thank you.